webinar with us, with the Global Plant Council. Please pay attention. As you can see, you can use freely the chat in order to introduce yourself, chat with each other, and make comments. But if you want to ask questions to the speakers, please use the Q&A tool that you must see in your toolbar on Zoom. And we will pick the questions exclusively through this tool, because it can be very challenging to find them through the chat if it's very active. So this is a new webinar from the webinar series of the Global Plant Council together with Plant Biotechnology Journal. Today we have a very special moderator. We have Dominique Michaud. He is a professor at the Université Laval in Quebec, Canada. His research focuses on plant biotechnology and molecular farming. His work explores how physiological factors, particularly hormonal regulation, affect recombinant protein production in plant systems, such as Nicotiana benthamiana. And he is going to introduce us a very special issue as well. So, Dominique, the floor is yours.
Thank you very much. So if I can share my slide. Okay, you see my slides?
Yes, we can see your slides.
Okay, so let's start. Sorry for this. So thank you very much, Isabelle, and thank you very much to all of you for attending today. I understand that we have a very large attendance, a large crowd, so that's great. So thank you to the Global Plant Council for organizing this, to Plant Biotechnology, of course, to Wiley also, to hold this, for the opportunity to hold this webinar today. So today the topic will be the Nicotiana benthamiana Biofactory, which is among plant expression system one of the most, if not the most, widely adopted platform to produce proteins, and now also increasingly to produce organics. And for this reason, we felt at PBJ that dedicating a complete issue of the journal to this expression platform would be both relevant and also useful. So before giving my short talk on hormones, I will take a few minutes to present the special issue, and also to present some basics about the platform, because I know that several of you are familiar with the system, but others are not so familiar with the expression of recombinant protein, but maybe not with the Nicotiana benthamiana. So I will start with this. And first, a few words about the special issue. So you have probably seen that the January issue of the journal, Plant Biotech Journal, is dedicated to this topic. And so in this issue, you will find 28 papers, but in fact, in the special issue, the complete issue, we have a virtual version online on the journal website. We are at 38 papers, I think, and a few others will be added. So it's a collective work of about 40 papers, which is very interesting. 40 papers, 40 excellent papers, including topical review on the different aspects of the platform. So it's been published very recently. And sorry, I will try to. Okay, so the content of the issue, it's about all aspects of the production platform. So first, we have 7 topics which cover the Benti agroinfiltration agrobacterium platform, up to the end, up to the downstream processing, purification of the proteins. So we have all topics covered in this issue. We have topical reviews on these different issues also, and of course, primary research paper, excellent papers, several excellent papers. So covering the platform, the interaction between the plant and the bacterium, transgene expression, protein engineering, design, metabolic regulation, post-translational modifications, environmental condition also, and downstream processing. And also we have several interesting case studies where are described approaches to produce important proteins or difficult to express proteins, and also organics, metabolic engineering approaches. So it's a very broad topic on a specific topic, in fact, and it's nice to see the participation, the contribution worldwide to this issue. So we have about 40 contributions, and we have contributors from 17 countries on all 5 continents. And so here you see the world distribution for the contributions, and here for the labs, for the teams participating. So overall, we had 40 labs contributing to the special issue, and overall more than 250 authors. So thank you to all contributors. I think it's been great to work on this for the last 1.5 years. And most importantly, also to thank the reviewers from everywhere for this special issue, because they have, of course, actively contributed to the quality of this. So I thank you, and I had the pleasure to co-edit this with my colleague Stephen Stretfield. So it's been great, and now we are happy to have this virtual issue published. So just before starting my talk, I will take a couple of minutes just to come back to the procedure itself. As I said, most of you probably are familiar with the procedure, but for several of you, maybe not, because you are possibly using this as a model platform for your science. But for the technical aspects, when you want to produce proteins or organics, I will give you a few words now. So it's quite simple, actually. So we have the seeds, and then we put them in soil, in the substrate, and then we transplant, and then we grow the plant for 3-4 weeks until they reach, let's say, 30 grams, 35 grams, 40 grams. So these are not transgenic lines. These are wild-type plants that will be used then for the agroinfiltration. So we then infiltrate the plants with the agrobacterium gene vector, these plants here. And there are several ways to do that. So we can do on a small scale. We can do it with a syringe, or if we want to express the proteins or to infiltrate all the plants, we can use a vacuum system. And if you are a company working in company, you maybe have robots also to infiltrate several plants at the time. And once this is done, so you have done the infiltration, so you have done the transfection. In fact, the bacteria will transfer the gene or the genes, and the plant will produce the recombinant products. So this takes about 1 week. So it's variable from 5, 7, 9 days, and then it's harvest, and then the easy job starts with the extraction, purification, eventually formulation. That's a big step here for the downstream processing. So this is a quick summary of the procedure, and so again, you can do this in the lab with syringe. This approach is used, in fact, for many studies, the basic studies in plant biology. Or you can exert a vacuum. So you put the plant upside down in the agrobacterial solution, and then after 1 or 2 minutes, you break the vacuum. So again, for people not very familiar with that, I will show you a short movie. This one from the Science TV magazine Découverte. So I have just taken some excerpt just to illustrate, just for you to visualize how we do this. So we put the seeds in the substrate, and then we wait. So it's horticulture at this stage for some weeks. It's high-tech horticulture, highly efficient horticulture, but horticulture. We put them in a greenhouse or in a grow chamber, and then we give them water, nutrients, and then when the plant is ready, let's say at 30 grams, we are ready to infiltrate. So here we have the agrobacterial solution, and then in this solution you have the bacterium, and it's a very simplified cartoon, but it's visual. So of course you have the TI plasmid with the tDNA, which is transferred to the nucleus in the plant, and then you are ready to infiltrate. So you can do this with a syringe on a small scale basis, but also you can use a vacuum. So So here in the vacuum, the goal is to remove the air, the bubbles, and then after let's say 1 minute, you break the vacuum, and then the solution will infiltrate in the leaf. You see here entering the leaf, and then it will take back the plants to the greenhouse or to the growth chamber, and the bacteria will do its job of transforming, and then the plant cell will do its job of expressing the product. So this was a short introduction on the special issue, and now it's my talk. So I will be talking today on hormonal balances as key determinants of recombinant protein yield and quality in Nicotiana benthamiana. So why hormones? Of course, hormones, you are familiar with that. It's very important for the development for many things, many physiological processes in plants. Why hormones? If we are talking about the benthamiana platform, it's because we can eventually control the interplays between hormones. In plants in particular, you know that, for instance, you want a given response in the plant, like for instance, getting a very high yield by promoting the primary metabolism, and you know that some hormones will contribute to that, for instance, cytokinins. But you know also that cytokinins will do the job if they can, because you have all these interactions. So this is a a very simple model, but in many cases you have several hormones interacting. So again, let's say that you have a response here for cytokinins, for instance. You say, okay, I would like to maximize the yield, so this would be response 2, but another hormone is there which will negatively interact. So we show some antagonistic activity against hormone C. But promoting hormone B, which would cause or lead to another response, and in some cases interfere with hormone C. So why taking this into consideration with a system like benthamiana is because one advantage of this system for expressing protein is it is purely artificial. So the basic principles are biology. We need to understand the biology, and it's very complex. And this plant is a model plant to understand many things in plant biology. But at the same time, because it's artificial, we can make changes in the system. We can promote, we can interact, we can interfere with the interplays between the hormones. For instance, here, if our goal is to get this response here, promoted by hormone B, we could, for instance, boost the amount of hormone B, because it's the balance, is the ratio between the two hormones that will determine the final outcome. So, or what we can do is to repress the expression of hormone A. And it's the same here for this model here. So you can, for instance, boost the production of hormone C to get response 2, or you can repress the production of hormone B to make sure that here hormone A, which is blocking the action of hormone C, but promoting the action of hormone B, won't be able to do it, because here we have less hormone B. So this will increase the ratio. All is trying to shift the balance depending on what we want. And because it's an artificial system, we can play with that. So for repressions, for instance, we can use chemicals, synthetic inhibitors. We can use gene silencing approaches. We can use gene editing like CRISPR-Cas9 also to block some specific reactions in the biosynthetic pathway. Or if we want to boost, we can use chemical activators. We can genetically transform. We can even co-infiltrate, because when we do the infiltrations, we have bacteria that are borrowing the sequence we want to express, but we can use mixtures of bacteria with different constructs, and then to express helper proteins or to express RNAi constructs, for instance. So we have many options with this system that we can control. So for instance, I will just briefly describe 2 topics. So there are many that involve hormones and that we might want to control. So for instance, The first topic is the growth defense trade-off. We know that when we agro-infiltrate the plant will be strongly reacting to the bacterium, and the plant will be producing huge amounts of salicylic acid, and salicylic acid will have a negative effect or a tempering effect on the primary metabolic functions. So, of course, the system is efficient. So we will have interesting yields, protein yields, but maybe we could have more if we can find a way to temper the production of SA, to temper the defense reaction. And so I will give you examples that have been published along this line. So, and the other topic is the host architecture. So several hormones control the plant's form, and three in particular are involved. For instance, cytokinins, which favor bud outgrowth, and auxins and strigolactons, which promote apical dominance. And overall, these hormones in the plant will determine the shape of the plant. From a practical standpoint, it could be interesting to control this because, for instance, in benthamiana, we know that bud outgrowth would be very interesting to get improved yield because we know that young tissues, young leaves usually are producing larger amounts of recombinant protein. So we might want to control this. So technically, what we can do, of course, again, so if we want to have to control the growth defense balance, if we want to have to improve the anabolic cellular processes in the plant and to get eventually to get improved recombinant yields, one way is to boost the primary system. Another way is to block or to make the defense response less strong. So this is quite simple, actually, the principle. The same here, if we want more bud outgrowth to improve the cytokinin to auxin ratio, and then to improve axillary growth, to eventually get a compact phenotype, which is an interesting trait for many plants, and then eventually to have a more interesting protein yield, we can boost the synthesis or the presence, at least the presence of cytokinin to increase the ratio compared to auxins. Or an alternative is to block the biosynthesis of strigolacton in such a way as to decrease the apical dominance and to get rid of this negative interaction between the cells and the CK. So I will discuss these different approaches now. So for the growth defense trade-off, the goal is to temper defense response. And this is a paper published 2 years ago by Tech in Germany. And the goal was to silence the expression of two genes. So SABB2, involved in the SA, associated with SA production, and COI1 associated with jasminic acid production, which are the main two defense elicitors triggering defense responses in the plant. And what she has done is to express GFP or beta nerve growth factor, beta NGF here, transiently in benthamiana, and when at the same time transiently expressing these constructs here to repress the expression of ACBP2 or COI1, she has got these yields here. So we see here for GFP, control, for beta and NGF, the control, and you see that the huge increase in GFP fluorescence and the huge increase in the production of beta NGF. So that's interesting. It shows that if you temper, if you repress, or if you decrease the production of SA or JA also, it works, then you will have probably more resources, and it will help the plant to produce more protein. So because it probably will boost relatively the primary metabolism. This is another example, interesting example published in PBJ recently, last year. So here the goal was to repress again the production of SA or the interaction with NPR1. So many several reactions related to the plant's defense. So they have tried several gene candidates, and especially here, SIRC1, ICS here, so upstream of SA accumulation, and then NPR1 here. So what they have been doing is to use VIGs for silencing immunity genes, and they also had double mutants for NPR1. So again, what you see here, you have a control here. So if you repress SIRC1, NPR1, ICS, you see the fluorescence here compared to the control. So this is for the VIGs constructs, and here for the mutants, it's the same outcome. So that's interesting. So these are two very interesting examples of being able to temper the defense response in the plant. It's important, and it will happen, the defense response, because when you infiltrate, of course, it's a bacterial infection. So the plant will be reacting. But if you are able to decrease the intensity of the response, the net result is more photosynthesis or more primary metabolism. So these are interesting examples where you can achieve this by depleting the pool of SA. Another approach, which is interesting, instead of playing on the side, we boost photosynthesis. We boost primary metabolism. So this is an example that we have been working on a couple of years ago in collaboration also with our friends at Medicago. So what we have done is to take this TI plasmid here, which is in strain AGL1 that we use for the transfections, and to integrate a gene in the backbone, the plasmid backbone here, to integrate a gene coding for transzeatin. So the coding for transzeatin synthase, leading to the production of transzeatin, which is a cytokinin. And why doing this? Cytokinins are involved in many physiological processes associated with primary metabolism, cell division, of course, but also photosynthesis. And we know that cytokinins have a very strong positive impact on the synthesis of photosynthesis-related genes. So it is what we got here. If we add in the bacterial solution, we add just a small fraction of this bacterium now expressing the cytokinin. It's interesting because it will produce cytokinins, and the plant will detect this and itself produce more cytokinins. So you see here the huge increase of cytokinin, different forms of cytokinins in the plant. And as expected also, so what we see, if we use this strain, TZ strain here, we add this in the medium, then we get more rubisco, large subunit, a small subunit, so maybe 2 times, sometimes more. So you get more of rubisco, and not only rubisco, also other photosynthesis-associated proteins like light harvesting complex B here from photosystem 1, or plastocyanin in the electron chain involved in that for the production of reducing power. So, and we have other examples. So many examples of genes and proteins also upregulated by the presence of more cytokinin, which was allowed by the use of this engineered TI plasmid. And what's interesting also is that if we get this more photosynthesis-related gene expression, it's interesting because you change the balance between the primary metabolism and SA without altering SA levels. But it's not the case, in fact. What we see is we alter SA levels. So we have less SA indirectly because we have boosted the primary metabolism. And if you look at defense genes, for instance, SAGT, here, sorry, which is involved in the biosynthesis of salicylic acid, or connextrypsin inhibitor, or also the pathogenesis-related protein is a chitinase here. You see that if we are using the modified strain, adding the modified strain in the medium, we get less expression of these genes. So by boosting the photosynthesis here, we got a reduction of the defense response in the plant. Of course, it's important to confirm also that the yield will be there. So it's interesting. If, for instance, here it's a diagnostic IgG or a therapeutic IgG here expressed in the plant. And if you add this strain in the medium, then you have more. The yield is increased by 2, 3, 4-fold. So it's the same also for GFP. You see the fluorescence here. So this is for in white here. This is if you add the modified strain at different doses. So that's it for that was my examples for the defense the growth defense trade-off. And then the other example I will be talking about is the host plant architecture. So the host plant architecture is interesting when you consider using benthamiana for producing proteins. And the main reason is because if you are here at the top, you see the young leaves here. These leaves are more efficient than the plants here, the zone number 3 here, which is less efficient. But if you look at the biomass, you see that the biomass for the plant is mainly explained by this part of the plant, which is not very efficient here, you see, compared to the younger parts, which is not very efficient. So if we can find a way to increase axillary growth to get more of a young tissues, more organs, for instance, in this part, more axillary growth. So if you can find a way to do that, it's probably interesting to improve the yields. So here, the idea is to boost the presence, the ratio of cytokinin to auxin and trigalactone, considering that cytokinins are improving or have a very strong positive effect on the primary metabolism. And they are potentially interesting to improve the yields because here you see that the young organs are very efficient in producing recombinant protein. So just very simply what we can do is to spray the plants with synthetic cytokinins. So we've done this with 6-BAP. So this synthetic cytokinin is used for different goals in greenhouse setups, in greenhouse production setup. And when we spray this hormone on growing plants in the greenhouse before the infiltration, so we have little impact on the primary stems and leaves, but we have a significant positive impact on the axillary. So S is for secondary, so on the axillary leaves. So if you look at the biomass here, we see that for these doses here compared to the control, we have 20-25% more biomass, which corresponds essentially to young tissues. And because young tissues are much more efficient than the older one to produce on a cell basis to produce a recombinant protein. So this 20-25% increase in biomass translates in an improvement of 50 to 60 or 60% of protein yield. Here it was hemagglutinin 1 for the development of flu vaccine. So you see that here that if we boost here this part of the model, we get more yield because we promote axillary growth. Now, if we take a look at this side of the model here, so if the goal is, if we want to, the goal was to reduce the plant special footprint. So for instance, you know that this plant occupies a given surface, a given volume in growth rooms. And in some cases, and it's the same for many horticultural plants and agricultural plants also, in several cases, the compact part, the compact phenotype is an interesting trait for improvement. Eventually, for instance, to increase the plant, the cultural densities in the production setups. So how can we do that? And what we have done here is to consider the possibility of repressing by CRISPR-Cas9, repressing the accumulation, the biosynthesis of trigalactone here. Why? Because it would break here this negative interaction with cytokinin. So then cytokinins are involved, are promoting bud outgrowth. And if we promote bud outgrowth right from the beginning, just when we put the seeds in the soil, it's quite possible that we get a compact phenotype. because we will facilitate improved bud outgrowth while also interfering with apical dominance. So here is the biosynthetic pathway for strigolectans here. And we know already that if we block this here, this enzyme, or this enzyme, so CCD7 or CCD8, these are two dioxygenases. We know that if we block this, we don't have prolactin, which is the precursors of strigolectin. So we have developed CRISPR constructs for repressing these genes. And here is quickly an image of what we got. So here on the left, you have the wild type plants. And then you see the mutants. These are independent mutants with different mutations. And you clearly see that they are more compact. What is interesting is that they have the same biomass. So you put the seeds in the soil, after a couple of weeks, after 3 weeks, it's the same biomass, but the general architecture is modified. The plants are much more compact. If we remove the leaf of this, it's even more convincing. So here you see this, the stems. So you have the control. So this is the normal situation, the wild type. This is the lab strain that everybody is using in labs worldwide. And these are the mutants. So you see here that the axillary stems start to grow right from the soil surface. So the phenotype is really changed. And then if we look at the spatial footprint, the volume occupancy of these plants, you see the difference here. So if you take into consideration the numbers we have, so this is schematically the occupancy for the wild type. And if you compare for the different mutant lines here, which lack strigolectin, you see that, for instance, for the red line here, it occupies less space. And more specifically, it's about 50%. So the volume occupancy of these lines is about 50%, which could be quite useful in, for instance, in vertical farming setups, if you could put more plants in the space you have, the restricted space that you have. That's interesting if we look at what happens in the plants. So you have the wild type and two of the lines, the red and the purple lines. So if you look at the auxins, it's the same amount, which is expected because it's upstream here. We have blocked this strigolectin accumulation. But if you look at the cytokinins, so we have a significant increase. So then we change the ratio as was the goal, actually. Of course, it's important to demonstrate that these plants, even though they have the same biomass, they are efficient in expressing recombinant proteins. So this is the example for GFP on a specific basis, weight specific basis on the plant, the total plant, also the yield. So you see that they are similar. So they are comparable in terms of efficiency to produce recombinant protein. And this is another example here for Rituxan. So it's a therapeutic antibody. So we have no negative impact. So that's good, because the first goal was to get plants that take less space, that are more efficient in using the space available. So it works. Now we're trying to improve the yield of that, because we have more axillary stems, more axillary leaves. So potentially, maybe we could get more interesting yields. So we're working on that now. And so I will stop here for these plans, because next month it will be the ISPMF meeting, and I will give a talk dedicated to that. So of course you can go to see this paper in bioRxiv that will be published soon in PBJ. So if you are interested. So I'm almost done. I'm done, actually. So I've discussed, the goal was to discuss the importance of growth hormonal balances in plants for many physiological processes, and how we can try to control this, to change this in the plant for practical goals. So I've discussed the case of the growth defense trade-off, of host architecture also. We've been working also on the senescence trade-off, because when you express large amount of certain proteins, these senescence reactions are activated, and we have shown recently, it's in PBJ, in the special issue, in fact, that if then we express constitutively a gene involved in the primary metabolism that was reduced by the expression of the production of ethylene, then we can reverse the situation and increase the yields. And also, we've been working on protein, endogenous protein load, for instance, during the downstream processing. Rubisco is a big problem in many cases. So, but we want to have a very good yields of photosynthesis because we want the protein we are looking for. But this rubisco is a contaminant. It's a super abundant protein in leaves. So if we could find a way, in fact, we have published on that, find a way to increase the ratio, to increase the amount of jasminic acid in the plant, which is a strong repressor of photosynthetic gene, we could maybe get some rubisco depletion before harvesting the leaves. So many possibilities. I'm sure you can imagine possibilities on your side. So I think I'm done for now. So I will just take a few seconds to thank all the contributors to these works over the years. Also, I had a very close collaboration also with Louis-Philippe Hamel and Marc-André Déo and their colleagues also at Medicago. Some of them are now at RMS Biotechnology. So we've been working together on these things for many years. And more specifically also, postdoc at Denver working on the plasmid engineering, Beatrice, Louis, and Christina working on the compact plants, and Marie-Claire Goulet, my lab manager. So thank you very much. Okay, so our next speaker is Frank. So Frank, in the special issue, has published a review paper, interesting review paper on the different approaches we can use, protein engineering approaches and different strategies we can use to improve, to boost the yields, to improve the quality also of benthamiana, expressing recombinant proteins and peptides eventually, and also organics. So Frank will be now giving us his talk.
Okay, thank you, Dominique. I will pull this up. Okay, so hopefully you can see everything and that you can all hear me very well. Thank you for the opportunity. Thanks to everyone for coming along this morning or today or tonight, depending on where you are. Like Dominique mentioned, we contributed a review paper to the special issue on the Nicotiana benthamiana plant factory, and it looks at the themes in protein engineering. I'm going to talk a little bit on some of the major themes for protein engineering approaches to increase recombinant product accumulation in Nicotiana benthamiana, and then focus a little bit on some future work done by an excellent PhD student who is now Dr. Max Harding, who was my co-author on the review, because it follows on from some of those themes. So I don't have to do much to introduce transient expression in Nicotiana benthamiana, just to say that we've heard a lot already about the scalability, and I will talk a little bit about flexibility and also adaptability. And that means to say that you can do a lot to the plant when you're doing transient expression, a lot of things that you might not be able to do to a plant if you're doing stable transformation. So you can be quite rough with it, and you can sort of get it to do a lot of things for you and make some complicated proteins. In general, we use elements from viruses to drive expression, and Dominique's excellent video showed we use agrobacteria to deliver that. And it's quite easy to combine different bacteria and different expression systems that accommodate multiple protein cassettes to produce recombinant products that are made up of lots of components. So lots of protein components co-expressed at the same time. And we use that a lot. We make use of that a lot to generate complex products. It's quite simple to do this using the agrobacteria infiltration technique. And you can end up, depending on the protein, with quite high yields. So, as yields that look like they come from microbial systems sometimes. And as we've heard, it's very fast. You can go from sequence to protein within a few weeks. And so our focus is on expressing complex proteins. And in plants, in particular in Nicotiana benthamiana, you can do that quite well. So amongst eukaryotic expression systems, plants have proven to be very good at expressing complex proteins with high structural fidelity. So they match the proteins that are produced elsewhere. And so these include enzymes, antibodies that I'm sure we'll hear a bit more about. And my particular interest is in virus-like particles. So making the capsids of viruses in plants. There's many reasons you might do this, and there's a long history of doing this. And so you can produce vaccines based on capsids, virus-like particle vaccines. So you can use a presentation platform, for example, this one based on Cowpea mosaic virus, to present epitopes as a vaccine. Or, of course, you can make capsid VLPs or enveloped VLPs corresponding to the virus that you want to protect against. And the main reason to put this here, because I'm not really going to talk about vaccines, is just to show that these are structures derived from plant-based expression. And so you can see that the structure that gets produced is very much in line with the expected structure of these viruses. So again, with the high structural fidelity. Then in our review, we touched on quite a lot of different protein engineering strategies, far too many to get into in this short webinar. But one of the main themes was around the different environments within a cell and how we can either improve them or use them or avoid them or just consider them in our recombinant protein expression design, experimental design. So targeting proteins to a different subcellular environment can either be used to mimic their natural expression pathway, for want of a better word. For example, secreted enveloped virus-like particles need to be secreted to get to the plasma membrane, and in plants you can do that quite well. There's been a lot of historical work showing that you can improve the accumulation of antibodies in different compartments of the cell, in particular the endoplasmic reticulum. And you can do things to modify the different subcellular compartments in order to improve the accumulation of your recombinant protein. And this is more the theme of what I'm going to talk about, how different subcellular environments affect your protein of interest and how you can create new subcellular environments. So in this example, in this figure, there's the creation of a biomolecular condensate to improve accumulation of a recombinant protein. And so there's just two examples that I want to highlight that sort of took us down this path quite a long time ago now. One of which was some work that I actually did with Dominique Michaud here and others to modulate the pH of the secretory pathway in order to improve the accumulation of acid labile proteins, in this case, recombinant hemagglutinin. The protein in this case was susceptible to the environment in which it was being produced. The other example was published at a similar time, but done a bit later. And it was an example where we've changed the subcellular localization. It did have the effect of improving expression of this protein, which is the capsid protein self-assembling into virus-like particles of murine polyomavirus. But what we also saw that it greatly reduced the toxicity. So steering the protein away from the nucleus where it was naturally going, gave us more recombinant protein, but it also greatly reduced the toxicity to the host. And so in this case, the recombinant protein is the problem. And so we start to work on different ways where we can create environments to host proteins, either to protect them from the host or to protect the host from the recombinant protein. So it's not a trivial task. All our pictures, I like to show this tweet that once upon a time caught my attention. The intercellular environment is far more complex than what we see in most diagrams. So the protein concentration inside an E. coli cell in this case here is about 200 to 300 milligrams per milliliter, which is much more than what is depicted in this diagrammatic depiction of a cell. And it's also much more than you can achieve in a test tube. So it's quite out of the range of what we're normally imagining proteins to be in. So our approach to creating new environments, this being a protein engineering talk, is to create protein-based compartments. And we use the protein shells of viruses, so virus-like particles. In this case, not as the product themselves, but as a cage to host the product. We have done this in yeast to protect the cargo to great effect in the case of an enzyme, but we're also doing this to protect the host. So expression of toxic products inside a cage to protect the host. So what are virus-derived protein cages? Well, they're virus-like particles. They're evolved as containers. You can produce them when they self-assemble. They are very, very consistent when they assemble, and you can produce them in various hosts. So what we can see is that you don't need the source host. It's not that you can only make plant virus-like particles in plants. You can make the virus-like particles from mammalian viruses or even bacteriophage in plants with no problem. And there are lots of different ways to load the cargo. So why would you do this? Well, I was not only is one of the reasons what I was just getting into about sequestering a recombinant product. We also do this to potentially have therapeutic delivery, biocatalysis, as I mentioned, for enzyme encapsulation, and for fundamental structural virology. So you can produce particles this way in order to determine their structure. This figure shows all of the virus-like particles that have been used up until the date that we published this for the encapsulation of recombinant proteins inside them. And the green ones, as far as I know, are the ones that have been used for that purpose in plants. So quite a few options. And you can see that tends towards the bigger ones, and that's because, well, we've done 3 of these, and it's because we want to have more cargo encapsulated. All right, so it's actually quite a simple process to extract these out of plants, and doing so you can get a very detailed analysis of the particles that you make. So this is blue tongue virus core-like particle, which is made of 2 proteins, VP7 and VP3. And through direct fusion to VP3, you can encapsulate proteins. So for example, GFP, you can see VP3 is on the inside, and direct fusion of GFP to that will make virus-like particles like this. They look exactly like the unmodified particles. And if you take them out, you can put them on a gel, and you can see that the ones that should have the GFP inside them are fluorescent. And if you do a bit more detailed structural analysis, you can actually see density for where the GFPs should sit. So it's on a flexible linker here, so you don't get a clear structure of that, but you can actually see that the GFP is inside the virus-like particle. And so that was our first sort of attempt to modify these, but it's a little bit limited through direct fusion because you can only accommodate things of certain sizes. and you have to have every single coat protein fused to your cargo. So that limits control over the loading capacity. And so we turn to another virus-like particle, and I'll just put it up here. That's the bacteriophage P22 virus particle in sort of scale with the interior of an E. coli cell. It's been used for a lot of this kind of work, but mostly for in vitro analysis of proteins that are encapsulated. And in this environment, we're trying to recreate the selectivity and specificity of encapsulation. With the Bluetang core particle, it was a direct fusion to the protein. So if it assembles, it's going to be encapsulated. But in this case, we encapsulate the protein by a fusion to an interacting partner. So that interacting partner is called the scaffold protein. And we fuse the cargo protein to that, and it will encapsulate as the coat protein assembles. The scaffold protein will interact with the interior surface of the VLP, and the cargo that's fused will get encapsulated. And actually, coincidentally, inside, you can get encapsulation of proteins at a concentration that approaches the concentration inside a cell. So it's quite an efficient self-sorting mechanism that can exclude a lot of host cell components and include just cargo protein. We optimized a lot of this in E. coli. So this is work that was done in bacteria. We tested a whole lot of different parameters and loading approaches to sort of optimize our loading. And we learned a lot from that. We were able to produce toxic proteins inside the virus-like particle in E. coli. And so from those studies where we showed that we could protect a host cell using this approach, we learned a few things along the way, like the fact that the charge of the cargo can actually be tuned. If you modify the charge, you can actually use that to tune the loading, which can be helpful because it helps you produce virus-like particles that are more consistent. And in doing this in E. coli, we showed that the protein itself was protected from proteolysis in the host, and that the recombinant product was no longer toxic to the host. So it was quite an interesting approach. And I want to shift to our sort of more recent unpublished results doing this in Nicotiana benthamiana. We've actually been able to make these bacteriophage virus-like particles in plant cells without too much fuss. It's actually quite efficient production. It's efficient as they assemble. We can see that nearly all of the protein that's produced is assembled, and that the cargo loading proceeds as expected, even though it's in the eukaryotic host. And what was nice about what Max Harding, who I mentioned at the beginning, did with this project is that he showed that we could co-encapsulate multiple cargo proteins. So it gives a great flexibility to what can be inside the virus-like particle. And so in using two different fluorescent proteins, you can see that when you have the red and the green together, you see a sort of yellow signal from the leaves where that's being expressed, and a yellow signal from the gel where you run the virus-like particles out on the gel. And so the co-loading is quite easy to achieve. Again, using that flexibility in transient expression in Nicotiana benthamiana by mixing constructs with the agroinfiltration, you can sort of get a lot of different components to interact in different ways. And so with this approach, Max has shown that you can load via the scaffold protein mediated fusion, and in going back to direct fusion, you can also load small peptides inside P22 VLPs by direct fusion. And I can't really show at this point, because it's unpublished, too much of the details, but it does permit the accumulation of these highly charged and otherwise toxic peptides, just as it did in E. coli. So instead of real pictures, I've got this little cartoon which shows that when you have the peptide on its own, your plant does not look happy. And when you encapsulate it and you're protecting, in this case, the host from the product, your plant does look happy. Interestingly, some of the same peptides that were susceptible to proteolysis in bacteria were not susceptible to proteolysis in plants, but that might have been because they were so toxic to the plants that the plant didn't have enough time to respond. Either way, by taking this approach of creating a new sort of subcellular environment to sequester the protein of interest as a quite, I don't want to say complex, but I suppose it's a fairly advanced protein engineering approach, but it's afforded us a lot of flexibility in what we can express in plants. And some of these peptides have great potential in cancer therapeutics as well as pesticides and all sorts of applications. So I don't want to go too much longer. Thank you very much for listening. I'll bring attention to Max and my review, which goes into a lot of the themes that I've talked about here, as well as a lot more detail on some of the more traditional strategies like creating fusion tags and things like that to aid in recombinant protein expression. Thank you very much.
Thank you very much, Frank, for an excellent talk, as usual. So our next speaker will be Kathrin Göritzer. So Kathrin is working at BOKU in Vienna, and she spent some time also in London, I think I understand, with Julian Ma. So she will be talking about very interesting topics as well. Kathrin has contributed two excellent papers in the special issue. One was an original paper describing, dealing with the engineering of the ER to accommodate recombinant accumulation of recombinant proteins in benthamiana cells. And the other one was a topical review on post-translational modification of proteins in benthamiana, which is, again, an interesting and very important topic as well. So, Kathrin?
Mm-hmm. So, thanks, Dominique, for the kind introduction. In my talk, we will now use the Nicotiana benthamiana factory and engineer it to produce Secretory IgA, which is a potential therapeutic due to its unique properties for mucosal applications. So until today, most of the antibodies approved in the clinics are of the IgG1 isotype, which is the predominant one in serum, is monomeric, has a long serum half-life, and also mediates strong effector functions, for example, through ADCC. It's, however, also susceptible to proteolysis, and so it is rather used for systemic targets. In contrast, Secretory IgA is the predominant antibody class in human mucosal surfaces, where it occurs as a dimer joined together by a joining peptide, and is further complexed with a secretory component, which is highly glycosylated, and this gives it high resistance to proteolytic degradation at these secretions. And this multivalency also allows it to show high virus neutralization and agglutination, and all these properties make it a good candidate to work in these very harsh environments and to be used for topical delivery. However, the therapeutic development of Secretory IgA has been hampered because of manufacturing difficulties. So in conventional mammalian expression systems, the balanced expressions of the heavy chain, light chain, joining chain, and secretory component is hard to achieve, which leads to low yields, incomplete assembly, misfolding, often suboptimal glycosylation, and therefore reduced functionality. And often also an additional in vitro assembly step of the secretory component is required, which adds cost and time. However, it was shown that Nicotiana benthamiana actually a promising platform for the production of fully assembled secretory IgA. So there we can use transient co-expression of the heavy light chain, joining chain, and secretory component, and in Nicotiana benthamiana, for example, the glycoengineered line. And then after 5 days, we can harvest the leaves and do affinity purification step, a size exclusion step that follows, and that gives us then around 50 milligram per kilogram leaf fresh weight of fully assembled secretory IgA. And this is already quite high compared to mammalian expression systems, like here human embryonic kidney cells, where you achieve around 1 milligram per liter. And however, the secretory IgA expressions still remains lower than IgG counterparts, and also quite varies between the antibody that is used, or the variable regions that are used. So there is still room for improvement to approach IgG-like yields. And so we can, for example, tune ER-centric levers to boost assembly and expression yields. So to our end here, we explored chaperone and co-expression. We had a look at glycooccupancy, and then also host engineering. And then later on, we will also try to elevate product quality and function through post-translational modifications engineering in the Golgi by modeling the O and N glycoengineering. And this can optimize stability, protease resistance, and other performance and qualities of IgA. So what are the limiting steps of secretory IgA assembly? So in HEC cells, usually if you co-express the heavy chain and the light chain together with the joining chain, this yields fully assembled dimer, although the secretory component incorporation remains challenging. However, in Nicotiana benthamiana, even if you co-express high amounts of the joining chain, it does not drive complete dimer formation. And so our conclusion is that the joining chain incorporation is a limiting factor for dimer formation in Nicotiana benthamiana. So in mammalians, secretory IgA assembly can be supported by ER-resident molecular chaperones, such as BIB, calnexin, carreticulin, or MCP1. And so we tried the strategy of co-expressing these factors, which increased total IgA yields up to 2.5-fold. However, it did not improve assembly efficiencies. So there's still a lot of monomer remaining. So we next targeted the glycosylation occupancy. We saw that in the IgA C-terminal tail piece, we find an glycosylation site that is under-glycosylated. And it was already shown that this glycosylation site is quite important for dimer, or for incorporation of the joining chain and dimer formation. This under-glycosylation happens during high expression of recombinant protein because the endogenous ER oligosaccharyl transferase is limiting. And however, you can overcome this by co-infiltrating a recombinant OSD subunit, for example, from Leishmanian nature. And there you can improve the glycooccupancy significantly, and this then also drives dimer formation. However, you can see that there is still monomer, or not fully assembled monomeric structure unit left over. So clearly there are some additional strategies that are needed. And so for this, we can take inspiration from nature. So antibody-producing plasma cells secrete antibody, a manifold of their own mass daily. And they handle this extreme secretory load by expanding their ER about 3-fold as they mature to B cells. From B cells. So we hypothesized that expanding the ER, also Nicotiana benthamiana, could enhance secretion and relieve ER stress and improve secretory IgA expression. So inspired by these plasma cells, we expanded the ER capacity by targeting the phosphatidylcholine biosynthesis pathway, which is the main membrane building block. And so we use CRISPR-Cas genome editing to target 3 endogenous CCT genes by inducing frame shifts between the catalytic and the lipid sensing domains, which rendered the enzyme constitutively active. So we generated 3 lines with 1, 2, or 3 homologs modified, and we also recovered a line that was overexpressing a truncated CCT1B. We saw that all 4 lines showed markedly enlarged ER with network densities nearly doubled. And that was driven by massive ER sheets, whereas we did not see an additive effect when more than 1 CCT gene was targeted. The overexpressing of a truncated CCT1B and a very strong viral promoter further increased ER sheets. But we also could see stunned growth and low biomass, which makes it unsuitable for antibody production. And so the gene-edited lines seem to be or offer the best balance of ER expansion and plant fitness. So we went ahead and tested the lines by expressing 2 different anti-SARS-CoV-2 Secretory IgA1 antibodies in wild type and the engineered lines. And we quantified the total and fully assembled functional Secretory IgA by immunoblotting and antigen sandwich ELISA. And we could see that across all mutants, the fully assembled multimeric Secretory IgA increased by up to 7-fold compared to wild type. However, the ER expansion not only raised the yield, it also shifted the product distribution towards fully assembled functional Secretory IgA. As you can see that here of the size exclusion chromatograms after affinity purification, where the major peak represents fully assembled Secretory IgA and the minor peak, the monomeric structure unit. And so, for example, for COVID-215, which already has high assembly efficacy of around 80%, it was further boosted to 87%. And in a poorly assembling antibody 2E8, this could be changed from 60 to 75% of assembly. To also gain further insights into the assembly dynamics and secretion pathways of Secretory IgA in these ER-engineered lines compared to wild-type plants, we perform subcellular fractionation by linear sucrose gradient to separate soluble from organelle-containing fractions. And fully assembled Secretory IgA predominantly localized in the soluble fraction, and smaller amounts appeared in the denser fraction containing the ER microsomes. Very notably, ER engineered line showed increased full assembly of COVID-215 and 2EA in the ER containing fractions compared to wild type. But which would suggest that we have a longer retention time in the ER of those engineered lines. Next, we also ask whether the ER expansion influences ER stress. And so first, with the empty vector controls, here white bars, the basal transcription of BIP, BCIP, and BDI comparable to it being vital and the CCT lines. And that would indicate that engineering the CCT followed by ER expansion does not trigger a constitutive UPR. However, upon Secretory IgA expression, both genotypes upregulate ER stress markers. And that was, for example, for COVID-215 with BIP rising 20-fold, and for BCIP and PDI 2 to 4-fold. And we observed a similar pattern for 2E8, although not so pronounced. So overall, this transcriptional stress response was not significantly different, though, from wild-type plants. So despite the similar UPR transcript levels, the protein levels told a completely different stories. So densitometry analysis of immunoblot showed that significantly higher amounts of BIP accumulate in the mutant lines compared to wild type, open expression of COVID-215. and also the similar length to E8. The same trend was observed for calnexin, however, not so pronounced as for PIP. So in some, CCT editing does not trigger a constitutive UPR response. However, upon secretory IgA expression, it induces UPR, and then we have a much higher accumulation of PIP. And this is a plausible explanation, or one of the plausible explanation is that the expanded ER volume provides a more dilute environment that limits PIP aggregation and enhances its steady state abundance. To then further dissect the role of ER chaperones in secretory IgA assembly and quality control, we transiently expressed exogenous chaperones with secretory IgA1. So guided by prior work showing that the ER chaperones can increase recombinant protein yields. We tested PIP, calnexin, carreticulin, PDI, and multiple combinations thereof. And the best single factors were PIP and calnexin from Arabidopsis thaliana, which markedly boosted both COVID-215 and 2E8 secretory IgA1, albeit with different extent. So our facet was that using ER engineering plus targeted chaperones, secretory IgA1 levels can approach 1 gram per kilogram leaf fresh weight, which is up to 20-fold increase compared to wild type without chaperones. And so our conclusions are that mechanistically, ER expansion likely enlarges luminal volume, reducing misfolding, aggregation, and extending the residence time for productive subunit engagement. And then while the chaperone co-infiltration further enhances folding and assembly capacity. So now we targeted we had a look at targeted ER engineering, which markedly increased yield and assembly. So now we do a shift towards from quantity to quality using Golgi engineering to fine-tune post-translational modifications. And as I already mentioned, secretory IgA is heavily glycosylated across all subunits, the heavy chain, joining chain, and secretory component. And many of its advantageous properties actually arise from these glycans. So we heard now already that the heavy chain N glycans support assembly, the heavy chain O glycans contribute to stability. The joining chain is also important for dimer formation, and secretory component N glycans play a very important role for protease resistance, pathogen interactions, and mucosal trapping and solubility. And therefore, to control these post-translational modifications is essential for biosafety and function. So if we start off now with O-glycosylation, so the human IgA1 has an extended proline-rich hinge region that carries up to 6 core-1 O-glycans attached to serine or threonine. However, in plants, this pathway is fundamentally different. So there, proline residues are first hydroxylated to hydroxyproline by enzyme class called prolyl hydroxylases, and then it's further decorated with pentose chains, which are likely arabinoses. And these hydroxyproline arabinoside modifications are plant-specific, non-human, and therefore undesirable for therapeutic IgA. So with CRISPR-Cas9 knockout of the plant prolyl hydroxylases that we identified are active on the IgA1 hinge region, and then through co-expression of Golgi resident mammalian O-glycosylation enzymes such as GalNAc transferases, we can replace the plant-specific hydroxyproline arabinolization with human mucin type O-glycosylation in IgA1. Of course, regulatory preference favors human-like O-glycosylation, yet I'd like to mention that plant-specific modification actually enhance IgA1 resistance to proteolysis across diverse secretions. So this kind of creates a design trade-off between clinical familiarity and maximal mucosal stability. So in contrast to O-glycosylation, the plant N-glycosylation pathway is broadly conserved with mammals, but the more limited Golgi enzyme repertoire yields comparatively homogeneous glycan profiles on plant-made glycoproteins. However, plant-specific modifications exist, and however, by knocking them out, like beta-1 xylose and alpha-1 core fucose, and then co-expressing again the respective mammalian glycosylation enzymes, we can achieve human-like N-glycans containing, for example, determinant galactose, sialic acid, or bisecting glucnag, or increased branching, or even polycellulation. And then for example, on example now of secretory IgA1, we can then produce secretory IgA1 with defined N-glycoforms. Here, for example, to produce a variant with the plant GNGN glycoprofile versus glycans which carry terminal sialic acid. And this then allows us clear structure function investigations. But controlled N-glycosylation also lets us tune proteolytic stability, solubility, or interactions with immunomodulators and pathogens. And so it also guides rational designs to design the most effective therapeutic. And so to wrap this up, I talked about how we can engineer the ER to expand the ER to boost secretory IgA yield and assembly. And we saw that this does not trigger a constitutive UPR, but extends the ER residence times and allows high accumulation of endogenous chaperones that assist in folding and assembly. And then if you combine the two strategies of ER engineering and chaperone co-expression, we can increase secretory IgA yield up to 1 gram per kilogram leaf fresh weight, which is quite an improvement from the 50 milligrams per kilogram that we started off with. We can also use targeted glycoengineering to program secretory IgA quality and functionality. So in the O-glycans, we can disable plant-specific modifications and co-express mammalian enzymes to achieve mucin-type O-glycosylation. And of note, these plant modifications but actually enhance protease resistance. We can also engineer the N-glycosylation pathway by removing plant-specific modifications, again, add mammalian enzymes to then install tailored N-glycans, human-like N-glycans. And these defined glycoforms do enable us to study and tune stability, solubility, and interactions with pathogens, for example, our immune receptors, which supports then the rational design of effective secretory IgA therapeutics. And with that, I'd like to thank a few people or a lot of people that have been involved in the research that has been presented here. So the Eva Stöger lab, the Richard Strasser lab, and the lab of Julian Ma, where the research that was presented here was conducted. So thank you for listening.
Thank you very much, Kathrin. Again, an excellent talk. So Isabella, I guess it's the time for the questions now.
Yes, it's time for the questions. Kathrin, can you take the slides down, please?
Yes, of course.
So we have a lot of questions here in the Q&A. Let's go with the first. Originally, I think it was meant for Dominique, but I think any of you can answer probably. Maybe you have different ideas. Instead of traditional Agrobacterium, can we use gene GAN for the transformation of Nicotiana benthamiana?
Yeah, probably. Yeah, technically it's possible. Not sure it will be as efficient. It's so easy to use Agrobacterium. It's so efficient, especially it's optimized for benthamiana. So So I would suggest that we all use agrobacterium. But yes, technically it's possible to use some other kinds of delivery systems. I don't know if Kathrin or Frank, you have something to add on that one.
I think I agree with you, Dominique. It is possible, but the flexibility and how fast agrobacterium is, I think you would always choose agrobacterium.
Yeah, I would choose it as well.
I concur.
It's easy, Frank.
Yeah. Well, look, yeah, it's the most efficient way
Mhm.
and scalable way.
Yeah.
Perfect. So I remind all the attendees to send their questions through the Q&A. So we are not going to pick up any question in the chat because it's going to be too challenging. There is too much written already. And we are not going to give the floor to attendees to use your microphone. So just keep that in mind. So next questions. This is a question for Dominique, but it's not here, which lead to me with the two genes they mean. How many homologs of these two genes you have used for silencing? Maybe
Okay,
you know.
So I guess the question is about the compact plants with the downregulation of the CCD7 and 8. So in benthamiana for CCD7, there are two gene sequences, but one is a pseudo gene. So that's a unique gene that we have been targeting. For CCD8, it was a unique gene. There's only one gene there. So it was kind of easy work, lucky. So just one for CCD7, one functional gene, and one for CCD8.
Mhm. Sometimes you are lucky.
Yeah, sometimes, yeah. So we chose the enzymes, and then we looked at the genome, and we're very happy.
So next question also for you. How can hormonal regulation be adjusted within transient expression platforms to optimize output while maintaining protein integrity, structural stability, and glycosylation profiles, considering the balance between stress reactions and synthesis?
Yes.
That's a long one.
That's an interesting question, and the short answer is that we need to try and see, and then to adjust. Because of course, when we describe the results we got and the positive outcomes we got, in several cases, it didn't work the first time. And so you're right. When we change, for instance, the hormonal profiles, you change many things in the cells. So, for instance, I think Frank was showing result where we change the pH in the secretory pathway. And while doing this, we have published the first papers, but then we published another paper just showing that the whole cell proteome was changed. And for instance, in the secretory pathway, the complement of endogenous proteases was changed. So we had to look at this because in some cases we realized that the changes were negative in practical terms of producing proteins, but in other cases it was positive. So and it would be the same for glycosylation and all the other processes, the stability, of course. So we need to try. It's clear that we need to do research not only for the basic concept we're working on, but also for what happens in the cell. Because when we are changing something in the cells, many things are changing. In many cases, it's very difficult to predict. So my answer again is let's see what happens and what happened, and then but in many cases it works, and also the plants are very plastic also, so they adjust, and yeah, so we need to look in detail in these questions.
Thank you. So next, I'm going to jump a couple of questions and go for one for Frank. So this one, I think so I'm going to read it. I think the original viruses open and release their cargo inside cells. But how can these self-assembly VLPs stay intact without releasing their cargo? Are these VLPs engineered, or is this a natural mechanism that these self-assembly VLPs stay intact? Thank you for the presentation.
Okay, well, thank you for the question. It's a good question. So it really depends on the virus. You know, there are some VLPs that some virus particles that actually don't disassemble to release their cargo. And the blue tongue virus core-like particle is one of those. It doesn't actually release its genome. It just releases individual transcripts through pores. But if you think about it, viruses in plants themselves are assembling inside the host cell. It's possible that some of those disassemble, but the vast majority don't, and they escape to infect the next plant. So I think that's more the case that you're getting at. Viruses that do obviously disassemble, and I think it's just a matter of it's a numbers game in a way. It really depends a bit on the virus, but I think it's basically that most of them stay intact, and then after a certain amount of time they might disassemble, but we purify them or analyze them before that.
Okay. Thank you. So, and I'm going to try to find one for Kathrin. I think this one on glycosidation is for you. Okay. As you know, glycosidation is a major problem in plant by knocking out psilocybintransferase and fucosyltransferase genes can give non-glycosylated plants. So is there any other glycosyltransferase gene that we need to knock out, or the double knockout is enough for non-glycosylated plant transgene generation?
Okay, so I find it interesting to say it's a major problem. We actually think that the plant glycosylation is an advantage.
Okay.
So because you can engineer it so well, it's so susceptible to engineering. So if you knock out the beta-1,2 xylosyltransferase and fucosyltransferase, you don't receive unglycosylated glycoproteins. You receive a glycan structure that is already humanized but simplified. If you want to have a product where N-glycosylation, for example, is important to produce the glycoprotein for folding, but you don't want to have an N-glycan on it. It also sometimes occurs for some viral glycoproteins, for example, to be more immunogenic. You could, for example, co-express an endo-H that cuts off the initial glycosidic structure that is added in the ER, and then you receive non-glycosylated glycoprotein. It's called the glycodelete method. So, yeah.
Thank you. So we have a lot of questions and not a lot of time. So I'm going to ask the speakers if they are willing to wait a little bit longer.
That's fine for me.
Fine for me.
Yeah, okay.
So
It's a bit late for Frank in Brisbane.
it's yeah, I know.
A little bit,
know.
that's okay. But I was expecting a little bit later. That's okay.
Okay, so let's see. There's a lot of thank yous here. Question by Maria Elena Goldman for Dominique, but maybe you can all answer. If you want to produce recombinant plant proteins, is it still advisable to manipulate plant physiological processes in an attempt to increase production?
I would say yes, we can, of course, because plants are very plastic. For instance, we have developed transgenic lines for expressing recombinant protease inhibitors to protect the recombinant protein in the secretory pathway. By doing so, we were expecting some problem with plant development because proteases are very important, but the plant is able to adapt to compensate by expressing other protease genes. So because of that, we've got the plants and it works. So and it was the case also for by controlling hormones. So for instance, we have changed the architecture of the Benti lines, the CRISPR lines, but again, the growth rate is the same. So the plant is adjusting. So even if you express plant genes, or even if you modify important physiological process, cellular processes in the plant, sometimes it could be toxic. So sometimes it could be problematic. Sometimes you could get very poor yields, but in many cases it works. But again, as for the previous question, I would say that we have to try because it's because we don't know in advance what will actually happen.
Okay. Thank you. Next question, I think it's for Kathrin, because of the time when it was sent. Firstly, thank you for the presentation. It was very insightful. My question is somewhere during the slides you mentioned about the hypothesis of increasing the space of endoplasmic reticulum. Will that increase the ATP and folding enzymes, or would it just create empty space?
So we hypothesize that it does not only create empty space, we think that it creates or allows for more space we could see that for common transcript levels of ER markers, they were not markedly increased in the engineered lines, but the protein levels of chaperones and other stress markers were significantly increased. And I think that is because there is more space. So there is less aggregation of PIP, for example. And so there is more available PIP or other chaperones to assist in folding and quality control and secretion.
Mm-hmm. Thank you. Next, it's also for you. Hello, currently I'm working on a plant-derived antibody against snake venom. So I want to know if there is any difference in stability and assembly of these antibodies in plants compared to the mammal ones, I assume.
So that depends a little bit on the antibody format you are expressing them. So if you express them as IgG, for example, then there are some differences to mammalian produced IgG, markedly the glycosylation. So if you produce them in wild-type lines where you still have the core-fucose attached, you could have reduced antibody mediated cytotoxicity. So reduced effector functions and also reduced half-life because you don't have the sialic acid and galactose capping. And also sometimes we observe in plant-produced IgG that you have underglycosylation of the CH2 resident N glycan, which is very important to maintain the open conformation of IgG. And so you could also have a portion of not fully functional IgG in your patch, for example. So yes, there are differences which are mostly related to post-translational modifications or glycosylation.
Mm-hmm. But will you recommend to try this on Nicotiana?
Yes, I would recommend trying that in Nicotiana benthamiana, especially in the glyco-engineered lines, because it has been shown that if you have full knockout of this specific core fucose, so if that is gone, then you have a multiple more effective therapeutic or active IgG. So that is one of the big selling points of Nicotiana benthamiana.
Mm-hmm. Great. So, and maybe we can finish with this last question, that it's a little bit for all. Also, it's specifically addressed to Dominique and Frank. What are the key regulatory challenges associated with the approval of plant-based biopharmaceuticals?
Maybe you can start, Frank.
Well, I think the main problem is just that there have not been many proteins or even classes of proteins from plants that have reached approval. And so I think that's still the biggest challenge, because apart from that, the challenges are the same as from any recombinant expression system. It's just that plants are a bit of an unknown. And so sometimes it's a matter of developing the requirements to meet regulatory approval, because they're unknown to a certain point. they need developing in order to then meet them. But aside from that, I think, yeah, it's just that they're a bit behind the game.
Mm-hmm.
Just so that's a very good answer, Frank. Thank you very much for doing it. And maybe we could ask also, you could ask people in the private sector also. They will have many things to say about that. It's I guess it's a bit complex, but it's worth trying it.
It's very complex and country specific, as far as I understand.
Yeah, that's the problem. In fact, it's the same for GM lines, for instance. It's very variable from one country to another, and so the rules are changing. So I guess I'm not quite familiar with that, but I guess that for the regulatory issues with these products, it's quite challenging because of this diversity of rules.
Mm-hmm. Yes, so there are still a lot of questions, but we are almost 10 minutes over the time. And there has been some requests from the people in the chat about getting your emails. So are you willing for me to share your email with the attendees?
Of course.
I'm asking the speakers. So
I am. It's very much findable online. Yeah.
Also, yes.
that is very true
So
as well, but I have to ask.
Mine on the chat.
Yes, so I thank you everyone for staying until the very end of the webinar today. Thank you all, especially all the attendees that have remained for until the very end of the Q&A session. As you know, or if this is your first webinar, you don't know, I will send you a follow-up email with the link to the recording that you will be able to watch whenever you feel like on YouTube. And I will also include the emails from the speakers in order for you to be able to contact them if you are interested. And I will also send you in the next week or so the certificate of attendance. So just that will take a bit longer than the follow-up, so just be patient. And next month we will have another webinar, this time with the Safe Cassava Network, and I think it will be on the 20th of May. You can check our website, the GPC website, the webinar calendar, and everything, all details are there. And I will also share some of this information in the follow-up. And regarding the speakers, thank you so much for this webinar. It was very interesting. It was people were very engaged, so that is always a luxury. And I think you had fun, and it was a good experience.
Thank you very much. Thank you to everybody for attending. Bye-bye.
Yeah,
Thank you.
It's my pleasure. Thanks, Isabel.
Yeah, thank you for the invitation.
Bye-bye.
Bye.
Bye, everyone.